Well, good morning, at least Eastern Standard Time, and I hope it's a pleasant evening in Europe. I want to thank you all for joining us. It's been a real pleasure chatting to folks who are going to be presenting and organizing this session. And I think we will have a tremendous time because of the breadth and depth of knowledge across the four individuals, the four speakers. All are working in epigenetics and transcriptional regulation of BAV and BAV-associated aortic disease. And they probably represent, I'll take a guess, they represent the body of knowledge that we should, that's world-class. Each speaker will have about 10 minutes to present their slides and make their presentation. And then we'll have about a 15-minute discussion at the end. So that's fairly straightforward. The goals of this webinar are obviously educational, but more importantly, to develop collaborations and knowledge sharing amongst the community. I'm sure that I think Lauren told me there were about 75 registrants for this session. I think that there'll be some very knowledgeable folks on board, as well as the speakers, and I think it'll be great to hear their viewpoint. We're going to start with Evaldas Girdauskas, and I always get that wrong, but I hope that's not too bad today, because he has to duck away after his talk for a few minutes, and then he'll be back at the discussion time. Evaldas has got his MD degree in Lithuania. He did his cardiac surgical training in Leipzig. Then did junior, I guess we can call it junior staff cardiac surgeon in Bad Boka. And then he was in Hamburg as full professor and co-director of cardiac surgery there for a good bunch of years. Now he's full professor and chair of Department of Cardiothoracic Surgery in Augsburg, Germany. And he's also head of the Mitral Valve Research Network of the Heart Valve Society, and has published widely, extensively, and knowledgeably. And I'm going to say that for all four of you. So, Evaldas, do you want to get underway?
Thank you, Simon. Thank you so much for. I'm just trying to share my desktop. Just a second. Okay. It should function now. Can you see my slides?
Yep.
Perfect. So, thank you so much, Simon, for this nice introduction. It's really great to be with you today. I know it's a very nice type of discussion using this kind of webinar, and I'm really happy to be a part of this. So I prepared a very short presentation, actually, just in order to stimulate the discussion afterwards. And I'm going to say a couple of words about microRNAs in a bicuspid aortopathy today. So very briefly, you all know what microRNAs are. These are basically small non-coding RNA molecules containing 21 to 23 nucleotides. And they are biological regulators of multiple biological processes in the human body. Predominantly, they function at the post-transcriptional level, and they are supposed to have many regulatory roles in the cardiovascular system. So the idea of using microRNAs is to implement them as circulating biomarkers in a cardiovascular disease. The main issue in aortopathy and in treating the aortic disease is the fact that the aortic size-based parameters are of very limited value, as you all know. Well, most of acute aortic syndromes occur in aortic sizes well below 50 millimeters, mostly in a gray size aorta between 40 to 50 millimeters. So that's where we need kind of an additional information, a kind of prognostic values using circulating or imaging biomarkers. And therefore, some of the circulating microRNAs have been evaluated as potential biomarkers. And some of them showed different expression patterns in bicuspid aortopathy. And a couple of them were analyzed in aortopathy patients and showed kind of relevant correlation with progression and the size of aorta and progression of aortopathy. On the other hand, we know that circulating microRNAs might be up or down regulated in a very wide spectrum of cardiovascular diseases, not only in the cardiovascular diseases, but in a kind of tumor diseases and inflammation, systemic inflammation diseases. And therefore, lack of a specificity is really a major issue in using them. And most concerning for us was the question whether concomitant valvular cardiomyopathy in bicuspid aortic valve disease might be potentially involved in the changing the expression patterns of target microRNAs. And therefore, in order to address this, we did this preliminary work, which summarized in this slide. And we looked and were asked by Simon to address this publication, this preliminary work, this publication, in my short presentation. And we analyzed here basically looked simultaneously at the expression patterns of 12 target microRNAs, which are known to be involved in aortopathy. And we analyzed before and we compared the expression in the plasma and the aortic tissue obtained during the surgery in around 50 patients in a cross-sectional analysis. And you see the kind of baseline characteristic of these patients here on the right side of the slide. The main finding was indeed that we were able to demonstrate a significant linear correlation between an expression of these target microRNAs between plasma and aortic tissue, at least for 4 from analyzed microRNAs. And this is only a hypothetic actually conclusion, saying that potentially circulating microRNAs may reflect remodeling processes in the proximal aorta in bicuspid aortopathy patients. And I like to finish my short presentation with the kind of translational outlook, looking at the potential directions and collaborations in this area. So what is the role of using microRNAs in bicuspid aortopathy? And I think that our knowledge on microRNAs are still extremely limited. And just based on the fact that there are more than 2,000 microRNAs and more than 300 baseline variables and cardiovascular manifestations in aortopathy patients, this problem and issue needs to be addressed by kind of big data analysis and major support by informatics. Another issue is that we're still limited in our knowledge regarding the specificity of microRNAs for aortopathy, and especially, as already mentioned, with regard to ongoing cardiomyopathy. This is quite prevalent in patients having a bicuspid aortic valve disease in terms of aortic valve stenosis or aortic valve regurgitation. And last but not the least, I think we still need, and it's a major effort, serial measurements to address the causality issue, instead of performing only cross-sectional studies that basically show only a significant association. So these are very briefly my thoughts about potential future directions and collaborations, which can be addressed in order to get forward with the issue of using microRNAs as potential biomarkers in aortopathy disease. Thank you very much.
Thank you, Evaldas. That was excellent and a very good framing of the issues in hand. So let's ask Maria Grazia Andreassi to go next. And Maria is a PhD researcher, research director for the Italian National Research Council at the Institute of Clinical Physiology in Pisa. She serves as Chief of Molecular Epidemiology and their Genetics Research Institute. So really, you've got quite some tasks there, Maria. And her main research interests concern the fields of molecular epidemiology and genetics of cardiovascular disease, including BAV, but also the development and progression of valve disease in general. She's also, I was just reviewing your CV the other day, Maria, and also interested in radiation-induced disease in healthcare workers,
yes.
and published extensively on that. And that looks very good. I'm looking forward to your talk. Thank you, Maria.
Thank you, Simon, for your presentation. I wanted to thank also the organizing committee for inviting me to this very interesting webinar. So today I discuss our preliminary and pilot study regarding the role of genetic polymorphism in macroRNA machinery genes in bicuspid aortic valve and associated arthropathy. So we have seen before that the macroRNA dysregulation may be a crucial role in the pathogenesis of MV arthropathy by modulating gene expression, inhibiting translation, or promoting the degradation of a target messenger RNA, regulating many cellular processes involved in the pathogenesis of a BMV arthropathy. And so, yes, the circulating macroRNA may be ideal biomarkers to predict the progression of the disease because they are very stable in fluids, including circulating fluids such as serum, plasma, and blood. And during the last years, several studies have documented the dysregulation of a macroRNA in different cases of BMV and arthropathy. Anyway, today there are no conclusive data for a routine application in clinical setting, in clinical practice for a panel of a macroRNA. So more research is needed in this field. So I briefly remember the biogenesis of a macroRNA. In the biogenesis are involved different protein and different protein complex, and including in the transcription, in the processing, in the export, and in the targeting. So polymorphism affecting the present in these genes, codifying for this protein, may modulate the macroRNA function, resulting in loss or gain of macroRNA function. And this polymorphism can be located in macroRNA genes, macroRNA machinery genes, and in the target. It's known that SNPs in seed regions of pre and pre-macroRNA and mature macroRNA are uncommon and unlikely to be important regarding a potential functional role. On the contrary, genetic polymorphism in machinery genes are more common and seems to have a functional role inducing changes in the production of macroRNA, and so leading to deleterious effects in the gene regulation. And during the last year, several studies have documented the association between this polymorphism and cancer risk and also prognosis that influence in the treatment in cancer patients. So we decided to investigate the association between these polymorphisms, 3 specific polymorphisms, in 3 macroRNA machinery genes, Dicer, Drosha, and exportin 5, to investigate the association with bicuspid aortic valve within a case control study. We included a group of patients, 177, and a group of controls of healthy controls with clinical evidence of a normal valve. As you can see, the demographical and the traditional vascular risk factors were not matched in this population. Patients had a ascending aortic diameter of 4.5 centimeter, and 54% of the patients had elective ascending aorta and aortic valve replacement surgery. We found a significantly difference in the genotype distribution only regarding the polymorphism of exportin 5, with a significantly difference with a reduced frequency for the minor allele, with a lower frequency of mutated homozygotes. At regression analysis, we found that this polymorphism was associated with a lower risk of BMV under both additive and dominant models, with a decreased risk by 48% in additive model after adjustment for confounders. Anyway, we didn't find any association with aortic diameter, aortic stenosis, or aortic valve insufficiency. What about the functional role of this polymorphism? This polymorphism is it is located in the 3' untranslated region of exportin 5 gene. And we know that in the messenger RNA, the 3' untranslated region contains the binding sites for regulatory proteins that influence that control the gene expression, and in particular, polymorphism in this region can alter the binding sites for macroRNA, can create a new binding sites or destroy a target sites. So resulting in alteration in messenger RNA translation, decrease and increase respectively. And we evaluated by informatic analysis the potential binding microRNA binding sites for this polymorphism, and we found that the polymorphism altered 9 microRNA binding sites. In particular, the presence of C allele induce destruction, destroy binding sites for macroRNA 499, and on the contrary, creates a binding sites for macroRNA 18A. And both these macroRNA are known to be important role in the cardiogenesis, and moreover, macroRNA 18A is a part of the cluster macroRNA 17 that it's known to influence the progression of aortic dilatation. So in conclusion and in summary, our pilot studies, of course, has to be validated in more larger population, and it's necessary to define the functional role of this polymorphism. Anyway, this study support the importance, suggested the need to study the role of genetic variants in macroRNA and machinery genes. In addition, multi-omics analysis maybe can be the most powerful approach integrating genome-wide and macroRNA expression data in combination with imaging to improve our knowledge and the opportunity to identify new biomarkers, circulating biomarkers, and in particular macroRNA biomarkers in EMV-associated arthropathy. Okay.
Well, Maria, thank you very much. Excellent talk, and I enjoyed the fact that what it got to the mechanistic export of microRNAs, a very important and different approach. Thank you very much. So let's move on to Steve Greenway, who is Clinical Associate Professor of Pediatrics at the University of Calgary. He's a pediatric cardiologist. His clinical training was completed at University of Manitoba, and then at Sick Kids Hospital in Toronto. He trained in the genetics of congenital heart disease in the Seidman lab here in Boston. And he and I met back then. But he says he's new to the field of epigenetics, but I think really that's not going to be a limitation at all. His laboratory focuses on translational research, and he's published extensively in this field. Steve, please feel free to go ahead.
Great. Thank you very much, Simon. And thank you to the organizers for the invitation to speak today. And I'm going to change gears a little bit and focus on cell-free DNA. And this work comes from our recent publication, where we were looking at aorta-specific DNA methylation patterns in cell-free DNA from patients with bicuspid aortic valve-associated aortopathy. Just a little bit of background. Cell-free DNA is created when a cell dies, and there's a variety of nucleic acids that are produced when a cell dies, but we've chosen to focus on cell-free DNA. And cell-free DNA is created during apoptosis as genomic DNA is cleaved into approximately 140 base pair sized fragments. And these pieces of genomic DNA are released into the blood and found in all individuals. Most cell-free DNA comes from white blood cells, but cell-free DNA from all organs that undergo apoptosis can be found in the bloodstream. And our lab first became interested in cell-free DNA from the work done by Snyder et al. Back in 2011, when they looked at sequence variants between donors and recipients in cell-free DNA as a marker of organ injury after transplantation. But an important advance that expanded the utility of cell-free DNA came about when it was recognized that cell-free DNA, in addition to retaining sequence variants found in genomic DNA, also retains epigenetic features of genomic DNA, specifically DNA methylation. And interestingly, we've shown that DNA methylation patterns can be identified that are organ-specific. And so there can be regions of DNA that are either hypo or hypermethylated, and these are unique to specific organs. And the examples of this are shown here, where you can see that, that at this particular site, it was unmethylated in brain relative to other regions of or other organs, and similarly for the pancreas. And then this group went on to show that levels of cell-free DNA arising from the pancreas were actually 100-fold higher in patients with diabetes compared to healthy controls. So we were interested in creating aorta-specific or identifying aorta-specific differentially methylated regions. And to do this, we used publicly available methylation data for a single aorta sample that was available. And we compared genomic DNA methylation patterns in the aorta to all other tissues and cells that were available. And we looked for regions of DNA that contain differences in DNA methylation, and we focused on small regions that could be detected in cell-free DNA. And we used the software package Medellin to bioinformatically identify 23 unique aorta-specific differentially methylated regions. And so with these, once we had our in silico DMRs, we went on to validate them in vitro. And to do this, we used genomic DNA isolated from 15 different organs, and we compared the relative methylation pattern at these sites in the aorta compared to other organs. And these results, unfortunately, showed us that the computational approach often overestimated the differences in methylation. And as for the 4 DMRs that we focused on, a subset of our 23 that we analyzed, you can see here that for chromosome 11, the methylation in the aorta is not as greatly different, from other organs as we might like. And similarly for the DMR on chromosome 22. This was shown here also for chromosome 18, where the methylation pattern in the aorta was very, very similar to that found in the brain. However, on chromosome 20, we could see that this region in tissue from the aorta was hypomethylated relative to all of the other tissues in the body that we surveyed. And so we then went on to correlate levels of these aorta-specific DMRs using blood from patients with bicuspid aortic valve associated aortopathy. And overall, we found that levels of the aorta-specific DMR did not correlate with aortic diameter. However, we did find that blood DMR levels did correlate reasonably well with regions with cell death in regions of increased wall shear stress as measured by 4D flow MRI. And so this data suggests that areas of abnormal hemodynamics are associated with increased levels of cell death, and that this injury can be detected in the blood using methylated cell-free DNA. So overall, we are optimistic about the use of methylation patterns in cell-free DNA as a non-invasive biomarker for aortopathy. However, our data to date is based on a single sample from an unknown region of the aorta. And given that there are developmentally unique regions of the aorta, we hypothesize that there's going to be unique differentially methylated regions between these different areas of the aorta. And then we could develop blood biomarkers for regional aortopathy and potentially distinguish between patients with dilation of the aortic root versus more distal disease. And so this project's been assisted by numerous collaborators as well as fantastic students. And I just like to acknowledge funding support from Alberta Innovates. Thank you very much.
Thank you, Steve. That was excellent. And finally, last but not least, we're going to hear from Calogera Pisano. Calogera got her MD degree back in 2007. And then completed cardiac surgical training at the University of Palermo and was a clinical fellow in cardiac surgery at Papworth in Cambridge. She then more recently completed a PhD in microbiology, immunology, infectious diseases, and organ transplantation at the University of Tor Vergata in Rome, where she still is as associate professor in cardiac surgery and has published really extensively on this topic, and I'm looking forward to hearing from her. Calogera, it's all yours.
I no longer see her. I'm going to see if I can find her.
I don't see her either. She was there a second ago. So as we say in the business, talk amongst yourselves. in the interim, Stephen, I'm going to ask you a question, if that's all right. We'll just carry on. Are you allowed to have a dog in the office, or is that okay?
I'm at home. I'm working at home. Yeah.
Good on you.
photo bombing our meeting. So yeah.
But more importantly, do you think that organ-specific miRNA patterns really are organ-specific, or could, you know, there's a lot of both cell type origins between brain and heart. Do you think there might be contributions or visible contributions from both?
Yeah, I think that's one of the many disadvantages of our approach, right? Is we take kind of a big chunk of aorta tissue and then isolate DNA from that. And you're absolutely right. There's going to be multiple cell types, represented, from that tissue. And, as I mentioned, we were using kind of a DNA methylation data off the shelf with no insight into where that sample came from. Was it, what part of the aorta? We've taken a different approach looking at the pulmonary artery, and there we are looking at cells specifically. So whether we're looking at pulmonary artery endothelium, smooth muscle, fibroblasts. So I think you can drill down a little bit and look, at cell-specific populations, and that could certainly be a potential confounder, right? It may explain maybe some of the correlation or lack of correlation that we see, is that we're not looking at the right cell type.
Yep. Calogera, it's good to see you back. We thought we'd lost you there for a moment. Can you hear us? I think we've lost your video.
The audio connection is still going.
Yeah. Calogera, can you hear us okay? So while Calogera is sorting out her Zoom or internet issue, Maria, a question, if I may. There's a lot of there's some data that says that XPO5 has a role in a couple of cancers. Would you expect to see the phenotype overlap between cancer and BAV, if there is any, to be reflective of the genotype?
But I think that I don't expect a particular link, but I think that the majority of the studies are only in the field of cancer. So it's important to verify the potential role in other disorders, in other disease, and also to define the functional role, because it's a very complicated to suppose that the same molecular mechanism or the same genetic polymorphism can influence different different clinical phenotypes and also molecular mechanisms involved in the pathogenesis of the disease.
Sorry, I was just sending a text, a Zoom text to Calogera. My apologies. So while she's sorting this out, I'm going to open up to questions from the audience, as it were, and please feel free to go ahead. Evaldas will be back in a minute, and if Calogera can join us, or the internet can work for her, then we will come back to her. and she can give her talk. So, open for discussion.
Anyone with questions can enter them in the Q&A box at the bottom of the Zoom screen. We'll be able to answer them that way.
Oh, I see. So the audience is muted, Lauren?
That's correct.
Okay, got it.
No questions yet.
No?
Could I ask a question for Maria?
Okay.
Yeah, Maria,
Of course.
you know, I don't know anything about microRNAs, but you know, the machinery seems that it would be so important and crucial. Do you think that would you expect wide effects from you know, SNPs altering the machinery? Do you think you might have non-specific or multiple changes? Do you think the machinery is too important, you know, to be altered?
I think our knowledge is very very little. It's very important because the machinery genes are relevant for the production of all macroRNA. So it's I think it's very relevant to define the functional role of this polymorphism in the machinery genes. So also in genes, in macroRNA genes, because it's very strange and not clear how the same genetic variant can influence the production of entire macroRNA. So in this field, I can ask to Evaldas. Can I ask him how it's possible to define a specific panel, because the majority of the studies conducted until now in the field of macroRNA demonstrated a very different, not concordant data among the studies. More than 200 macroRNA have been described to be associated with BMV and arthropathy, and so only a little percentage of, I think less of 10% of the data are similar among studies and similar in the changes in down or up. So it's very complicated to define a panel in these different results. What about Evaldas on this point?
Maria, I'm not sure if Evaldas is back yet, but I see Calogera is here. Would you like to give your talk? And I'll do an introduction first, if that's all right.
Yes.
Good. It's good to hear from you. So, Calogera, Dr. Pisano did her MD degree in Italy back in 2007. She then did cardiac surgical training in cardiac surgery in the University of Palermo, was a clinical fellow at Papworth in Cambridge, did a PhD in microbiology, immunology, infectious diseases, organ transplantation at the University of Vergata in Rome, and is still there as associate professor and in the cardiac surgery unit. So I'm very much looking forward to your talk. So please go ahead.
Yes. Thank you, everyone, and thank you to give me the possibility to be here this evening. Sec, my computer is not working very well. Okay, it's fine. We in our university we investigate a particular form of aneurysm, the familiar forms in particular associated with bicuspid aortic valve. As we know, aneurysm are the 18 most causes of death in all individuals and the 15 most common individuals older than age years. In particular, we observed in our center that most of aortic dissection involved patients with no dilated aortic ascending aorta. In our opinion, the diameter is not the only parameter that need to be considered in order to prevent aortic dissection, as the guideline advise. According to our opinion, other biomarkers need to be considered in order to prevent aortic dissection. And this was the reason of our study. We make a question: is the diameter of the aorta a safe parameter for cardiac surgery indication in the aortic aneurysm, in particular in patients with bicuspid aortic valve? Our studies demonstrated that the diameter is not a very sure parameter to give a good prevention of aortic dissection, as also the International Registry of Acute Aortic Dissection show. We perform a multidisciplinary study involved not only the cardiac surgery, but also the anatomical pathologist, the engineer, the biomedicine of Vergata University and Palermo University. The aim of our research was just to identify different histological phenotype of ascending aortic aneurysm, and in particular investigate and evaluate potential genetic targets involved in the sporadic aortic aneurysm associated with particular form of aortic valve, in particular bicuspid aortic valve, and identify not only the histological feature and genetic feature, but also the transcriptional factor involved in this form of aneurysm. Our population of study involved dissected patients and aneurysmatic patients. In particular, dissected patients' age was about 16 years, plus minus 9 years, and the aneurysmatic population was about 61 years, plus minus 10 years. The first step of our investigation was identified three different phenotype of aneurysm: aneurysm phenotype 1, phenotype 2, and phenotype 3. In particular, we identified different phenotype of aneurysm according to the grade of medial cystic degeneration in the media, and according to the inflammatory cell infiltration and the apoptosis, and also the concentration of metalloproteinases. In particular, we identified three different phenotypes of aneurysm. Aneurysm phenotype 1 was the type of aneurysm that show normal wall at the time of operation, unless of the diameter. Histologically, this kind of aneurysm was characterized by a cystic medial alteration balanced by a good fibrosis in absence of medial apoptosis and with a low concentration of metalloproteinases 9. The phenotype 2 show in theater moderate wall thickness, and histologically it was characterized by an higher cystic medial alteration that substantial fibrosis with focal medial apoptosis and moderate concentration of metalloproteinases type 3. Type 9. The phenotype 3 of aneurysm show at the time of the operation thin and weak wall. It was characterized by elevated cystic medial degeneration without good fibrosis, and we found a plurifocal medial apoptosis with a moderate and severe concentration of metalloproteinases 9. This kind of aneurysm, phenotype 3, was in particular associated with aneurysm and bicuspid aortic valve and with dissection. In our opinion, the phenotype 3 of aneurysm, characterized by a severe cystic medial degeneration, absent substantive fibrosis, plurifocal apoptosis, and severe metalloproteinases concentration, is the particular phenotype of aneurysm associated with the orthopathy in bicuspid aortic valve, and it is also the precursors of dissection type A pathology. For a genetical point of view, we try to investigate some kind of genes in patients with this phenotype of aneurysm. In particular, we investigate the genes of metalloproteinases 2, metalloproteinases 9, endothelial nitric oxide phase, angiotensin converting enzyme, toll-like receptor 4, CC chemokine receptor, and transforming growth factor beta. We identify a particular gene related to inflammation correlated with this kind of aneurysm. In particular, the toll-like receptor 4 was the first step to causes medial cystic degeneration in ascending aortic aneurysm, in particular in patients with a bicuspid aortic valve. We published this result in this paper in which we identified a particular isoform of toll-like receptor 4, the RS4986790 isoform, was characterized by patients with bicuspid aortic valve, usually female, and particular severe aortic ascending aneurysm. And in our opinion, the toll-like receptor 4 mediates the activation of a particular pathway, inflammatory pathway, that can cause the senescence of aortic, smooth aortic cells, in particular the cells in the median aortic wall, and that can cause arterial remodeling, medial degeneration, and the onset of the arterial disease. We identified a particular risk genotype related to the toll-like receptor 4. This genotype was characterized by this particular SNPs, single nucleotide polymorphism, related to the other genes. In particular, in this table, you can just see the SNPs number of the genetic mutation in patients with phenotype 3 aortic ascending aneurysm and bicuspid aortic valve with high susceptibility of aortic dissection. In our opinion, the patients that had this particular genotype is an high responder genotype patients for aortic dissection, independently of the aortic diameter. We identified in these patients higher levels of systemic plasma inflammatory mediators, for example, interleukin 6, TNF-alpha, protein C, MMF2, MMF9, and other inflammatory factors. The immunohistochemical quantification of lymphocytes permit us to identify particular subpopulation of T lymphocytes and macrophages in aorta simples of patients with ascending aortic aneurysm and bicuspid aortic valve. In our opinion, the activation of toll-like receptor 4 pathway can cause an higher activation of other cytokine in the blood, and finally it can cause an higher expression of metalloproteinase 2 and 9 that can cause the elastic fragmentation in the medial wall of the patients with ascending aortic aneurysm involved in dissection and bicuspid aortic valve. In these patients, we also study the length of the telomerase of the leukocytes, and we know that in these patients there is a low high telomerase activation. Also, patients with bicuspid aortic valve and ascending aortic aneurysm have particular deficit in endothelial progenitor cells, and he had a particular changed the population of endothelial progenitor cells that can cause a relative low fibrotic reaction in the reparative process of the aortic ascending aneurysm. Another important biomarkers for us was the red distribution width. We studied the rate of red distribution width in patients, in this kind of patients, and we show a high concentration of RDW, that is expression of inflammation in this kind of populations. An important role, in our opinion, is also associated with the transforming growth factor bed pathway polymorphism. In particular, the isoform RS900, transforming growth bed 2, is a particular marker of gender susceptibility in patients with aortic aneurysm and bicuspid aortic valve. In fact, is associated with a female gender in this kind of patients. We performed a study also related to the PDEA expressions, and we show a particular susceptibility of this gene in patients with ascending aortic aneurysm and bicuspid aortic valve. Finally, we try to identify the abnormality of flow and association of abnormality flow and genetic alteration in patients with bicuspid aortic valve. And we identify two different types of aneurysm associated with different phenotype of bicuspid aortic valve. In particular, bicuspid aortic valve with the right-left fusion is mostly associated with a root dilatation. On the contrary, patients with the right non-coronary bicuspid aortic valve show mostly a tubular ascending tract dilation. And our histological, genetic, and immunohistochemical study show that the patients with the root dilatation had the high risk phenotype aneurysm associated with a higher risk of aortic dissection, in particular type A. In this group of patients, aortic root dilation associated with right-left fusion, we found an high level of interleukin-1 bed, interleukin-6, and interleukin-17. And also particular subset of lymphocyte T associated with this kind of patients. Also in these patients, we identify and study particular subpopulation of endothelial progenitor cells. We identify particular shear stress different grade of aortic wall in different different kind of population. In this moment, we are involved in this particular study with the engineer of our university. This is a new protocol that we are just started in our university, and we hope that next year to show the result. In our opinion, there are two different theories that can cause aneurysm and dissection in patients with bicuspid aortic valve. There is an hemodynamic theory characterized by the blood flow abnormality associated with different valve phenotype, and there is a genetic theory associated with the Notch gene. The hemodynamic theory and genetic therapy can prove. Yes.
Dr. Pisano, I'm sorry, we're getting close to the end of our time. Can I ask you to wrap it up now?
Yeah.
And then we will proceed to the question and answer phase
Yeah.
for just the last remaining few minutes.
Yes, it's fine.
Thank you.
It's fine.
Okay.
Thank you too.
Thank you. Very nice. Enjoyed that. So question and answers. Maria, I don't know if Evaldas is back. And he may not have heard your question, Maria. Evaldas, are you there? No, like every good cardiac surgeon, he's been dragged away to the OR. So let's open up to the attendees. Questions, thoughts, big pictures. And just as a reminder, just type a quick message in the chat. Lauren, is anyone typing? Because
I don't see any questions at the moment, no.
No, nor do I.
Good.
Oh, God. We'll do 30 more seconds, and I think we'll wrap it up because it's 11:58, and at least
Yes.
here on the East Coast, lunch is close, and I'm sure in Europe, dinner is close. All right. Well, I want to thank all the speakers. Very nice. I enjoyed it tremendously. And I look forward to the next Marfan Foundation webinar. They've been really tremendous. I've been very impressed by the quality and quantity of talks and the science that's come out of it. So thank you all very much. Thank you to the speakers. Thank you for the attendees. Lauren, do you have and Joe, do you have anything you want to add before we sign off?
No, thank you.
No, thank you. Thank you.
Great. Thank you very much. Bye-bye.
Thank you all. Have a good day.
Thank you very much.
Good evening.
Thank you very much.
Bye.